Hi class, today we will learn about the psychology of brain and behavior. The nervous system is the body's electrochemical communication circuitry, and it is studied by neuroscientists. The nervous system is made up of billions of communicating cells. And fun fact, there are 50 million nerve cells in just 1 cubic centimeter of brain tissue. Plasticity is the term that describes the brain's exceptional ability to change. Thinking is a physical event, and our thoughts are reflected in physical activity that occurs in the brain. So changing the way that you think is actually rewiring your brain. Pathways in the nervous system. Our nervous system is made up of sensory nerves and motor nerves. Our afferent sensory nerves carry information to the brain and spinal cord, communicating information about our environment and internal body processes from sensory receptors to the brain and spinal cord. And our efferent motor nerves carry the nervous system's output out of the brain and the spinal cord, telling areas what to do. For example, telling our muscles to contract when we move. Most information processing occurs in neural networks. Those are interconnected groups of nerve cells that integrate sensory input and motor output. So for this next section, it may be helpful to define a nerve. So what is a nerve? Nerves are bundles of neurons that are distributed throughout the body that help make up our nervous system. There are two primary divisions of the nervous system: our central nervous system and our peripheral nervous system. The central nervous system, or CNS, consists of the brain and spinal cord, and it is the location of over 99% of all of the nerve cells in our body. And you can see it illustrated on the left in red. Our peripheral nervous system is a network of nerves connecting the brain and spinal cord to other parts of the body. So it connects our central nervous system to the rest of the body, our limbs. And it consists of the somatic and autonomic nervous system. It's illustrated over here on the left in blue. So the peripheral nervous system is divided into the somatic and autonomic nervous systems. Our somatic nervous system, sensory, carries sensory information from the skin and muscles to the central nervous system about experiences such as pain, pressure, and temperature, our sensations. The autonomic nervous system takes messages to and from the body's internal organs, monitoring important processes such as breathing and heart rate, digestion as well. The autonomic is further subdivided into sympathetic and parasympathetic. Our sympathetic arouses the body to mobilize it for action, so it plays a major role in our experience of stress, our flight or fight response. And the parasympathetic calms. It helps us rest and digest. The stress response. It's the body's response to stressors. Stressors are the events that threaten people and tax their ability to cope. Stress is a response of individuals to environmental stressors which may be acute or temporary, chronic or long-term, characterized by persistent autonomic arousal of the sympathetic nervous system, which is responsible for our body's fight or flight response. Those physiological changes the body undergoes in order to prepare itself to confront a threat. Like, for example, your heart races, your palms are sweaty, and corticosteroids are released, those powerful stress hormones which are beneficial during emergencies. I've included this excellent video that explains how stress can make you sick. It goes into detail explaining exactly how that connection really exists. It's very enlightening, so whenever you get a chance, please review that on your own time. Now we'll talk about the cells that make up the nervous system. There are two types. First, we have the neurons. Look, you can see in the illustration, it's made of a cell nucleus and a soma, which is the nucleus is inside the soma. Then there's another word for the soma, and it's called the cell body. But you'll see soma and cell body used interchangeably. And then coming off extending from the cell body are the dendrites, and they receive information from other neurons. And then you have the axon, that's like the big tail, and it's covered in myelin, which helps the neurons send messages really fast. And then branching off of the axon at the end are the synapses and axon terminals. And then at the very end of, right before the synapse, which is the space, there is the terminal button, which is what neurotransmitters come out of. And then the second are the glial cells. And the glial cells are the support system. They provide nutrition, support, and other functions to help keep the neurons healthy and running smoothly. So neurons are specialized cells. They're specialized for transmitting information. And they have a specialized structure. And you won't need to know all of the different forms of neurons, but I just wanted to show you in the illustration to the right that they're not all made up the same. They don't all look like the standard neuron, the multipolar that we see in textbooks. But anyhow, the dendrites at the top, those are the receiving. They're the projections from the neuron that receive information and transmit it to the cell body. Another way to differentiate between dendrites and the extensions coming out from the axon are that the dendrites are surrounded by the cell body or the nucleus. And the cell body contains like I said, the nucleus, which is the DNA or the blueprint for life. And the axon, that's the long tail looking thing, sends the information that is received from the dendrites away from the cell body towards another neuron, which would be the receiving neuron or the postsynaptic neuron. And the synapse would be that space that is just past the very end of the axon, past those extensions on the axon. And once again, the myelin sheath speeds that neural transmission. So it speeds the communication, speeds up the transmitting the information. Information transmission. I'm sorry. Sorry about that. Bear with me, please. Here's another very interesting video that I showed in class that you can watch on your own time. It'll serve as sort of an introduction to neurons for those of you who may not be familiar with the topic, or for students who are interested in learning a bit more. This short video is interesting for two reasons. It discusses neurons in a different species, and it also describes really cool new research involving rats, looking into how dendritic spines change shape to allow for stronger synaptic communication. Really interesting stuff. It's time for some neurotrivia. All right. About how many neurons does the human brain contain? Give you a second. You can pause the video. All right. Answer is 100 billion. Second question. On average, how many connections does the average neuron make with other cells? 10,000. Number 3. How long can axons grow, measured in feet? Up to 3 feet, from the top of the brain to the bottom of the spinal cord. And number 4. How fast does a neural impulse travel, measured in miles per hour? This might be my favorite one. My favorite answer was at the speed of light. That isn't true, but it is still very fast. It's up to 330 miles per hour. Very, very fast. So how do neurons communicate? How does a living cell in our body communicate electrically? When you're thinking about neural communication, it may be helpful to think of the axon as a tube encased in a semi-permeable membrane. Being semi-permeable means that fluids can sometimes flow in and sometimes flow out, with hundreds and thousands of tiny, typically closed gates, called ion channels embedded in it. Ions are electrically charged particles that float in the fluids inside and outside of that semi-permeable membrane. And those particles include sodium, which is Na+, potassium, which is K+, chloride, which is Cl-, and other anions, which anions, it just means negative ion. So when a neuron is not firing, it is said to be resting. That also means it's polarized, with more positively charged sodium and potassium ions outside of that axon tube, and more negatively charged chloride ions and other negatively charged ions inside of the tube. And resting potential is a term that describes this voltage created by polarization. And a polarized cell means that there is a difference in charge, negative 70 millivolts on the inside of the cell, where the chloride and other anions are, relative to the positively charged outside of the axon tube, which is where the sodium and potassium are. A depolarized cell means that there's no more difference in the charge, or that the difference is decreasing between the inside and the outside of that axon. So more sodium flows inside of the axon, making the inside of the tube more positive. And the difference in charge between the inside and the outside of the neuron is decreased. So when the sodium channels open and the cell is depolarized, an action potential sweeps down the neuron, meaning the neuron is firing. And the action potential, that's that brief wave of electrical charge that fired down the axon. And it follows an all or nothing principle. So once the electrical impulse reaches the neuron's firing threshold, which is negative 55 millivolts intensity, the neuron fires all the way down the axon as if a fuse had been lit. And it follows saltatory conduction, meaning the signal jumps from node to node. If you go back and look at an axon, you'll see the myelin sheath is covered in, it looks like these little nodes. And there are spaces where the myelin sheath, it seems to be broken, and it sort of jumps across. So I think that's very interesting. And I hope you do too. So here's a graph of an action potential. The voltage is on the y-axis. To the left, it is the vertical axis. And you can see thresholds at negative 55 millivolts. And number 1, there is the stimulus. And you'll see a couple failed initiations. It does not pass the threshold of 55. But once a stimulus does pass that threshold, a action potential is fired as if a fuse had been lit, and it goes up. And then number 2 is depolarization, so more sodium ions in. And then at the very peak, it goes down, potassium, sodium goes out. There's repolarization, and then it goes underneath. There's hyperpolarization, goes back up to resting rate at 5. And resting rate is negative 70 millivolts. Here is a really excellent video illustrating the electrochemical gradient that I think will help you understand this material, because it can be complicated, particularly if you haven't taken biology yet. So I hope you find it helpful. All right, chemical transmission. So if neurons do not touch and electricity cannot travel in the space between them, how do they communicate? This is where the part of electrochemical communication comes in. Synapses. So at the synapse, this is a site where chemical communication between neurons occur. But it's not necessarily a site so much as that it's an absence of, it's a space. So it is the gap between the terminal buttons of the sending neuron and the dendrites of the receiving neuron. neurotransmitters are chemical substances that transmit information across the synaptic gap to another neuron. And the terminal buttons are those knob-like structures located at the ends of axons, and the synaptic vessels are tiny sacs within those terminal buttons that store the neurotransmitters. Synaptic transmission. So please watch this video of neural communication to see how many you can locate. And the receptor. So how receptors work. If the shape of the receptor site corresponds with the shape of the neurotransmitter, then the neurotransmitter will act as a sort of a key, opening the receptor site and enabling the postsynaptic neuron to receive the message sent by the presynaptic neuron. So postsynapse means the neuron after the synapse, and the presynapse means the neuron that's sending before the synapse. And reuptake. So reuptake. This is when some of the neurotransmitter is reabsorbed by the axon that released it, stopping its activity. And a lot of you may have heard of SSRIs. They are selective serotonin reuptake inhibitors, and they prevent, and how they work is they work by preventing that reabsorption of serotonin. So keeping more serotonin in that synapse to bind with neurons. So neurotransmitters. So this is not a comprehensive list of neurotransmitters, but some of the most commonly discussed in psychological science. So first we have acetylcholine, and that's for muscle control, learning, and memory. And GABA, it is an inhibitor, so the inhibition of action potentials, so it inhibits the firing of neurons, and it's also important for anxiety reduction. Glutamate is an excitatory neurotransmitter, so it is involved in the excitation of action potentials. It's important in learning and memory. And norepinephrine for arousal, vigilance, and attention. Dopamine, your voluntary motor control, reward, and motivation. And serotonin, emotional states, impulsiveness, dreaming. Endorphins, pain reduction, reward. And oxytocin, love, important for love and social bonding. So this is a fun little animated video by the Amoeba Sisters that sums up what we have learned so far. To skip to the summary, you can start the video at 10 minutes and 6 seconds, and I highly recommend you watch it. It's great. It really is great. So we've made it to the second major part of this lecture. Thanks for hanging in there. We're now on to the structures of the brain and their functions. The brain is organized into three major parts. First, we have the forebrain, the brain's largest division and most forward part. Next, we have the midbrain, which is between the forebrain and the hindbrain, and is an area in which many nerve fiber systems ascend and descend to connect to the higher and lower portions of the brain. Specifically, it relays information between the brain and eyes and ears. The hindbrain is located at the skull's rear, the lowest part, which consists of the medulla, cerebellum, and the pons. The brainstem is a very important part, and it includes much of the hindbrain and midbrain. Hindbrain structures include the medulla, which is important for breathing and heart rate, the pons for sleep and arousal, and also the midbrain structure for the reticular formation, which is stereotyped patterns of behavior. The limbic system has two principal structures, the amygdala and the hippocampus. The limbic system is a subcortical, which means under the cortex, the cerebral cortex. It's a subcortical network of structures important for memory and emotion. The amygdala is important for discrimination of objects necessary for survival, emotional awareness, and intense emotional expression. The thalamus is the central relay station of the brain. It's analogous to a server in a computer network. The hypothalamus, hypo meaning under the thalamus, monitors 4 activities known as the 4 F's: fighting, fleeing, feeding, and mating. The cerebral cortex. It is the wrinkled outer layer of the forebrain that is responsible for our higher cognitive functions such as thinking, learning, and consciousness. The regions labeled in white are the lobes of the cortex. The blue labels are part of the hindbrain. It's important to remember that there are 2 hemispheres of the brain, so this means there are 4 lobes for each of the hemispheres. So in this picture, we're looking at what would be the left hemisphere. So there are 4 lobes for the left hemisphere, and there are 4 lobes for the right hemisphere. So the frontal lobe is responsible for movement, behavior, problem solving, and speech. The temporal lobe is responsible for memory, language, hearing, emotion. The parietal lobe is responsible for functions of intelligence, language, sensation, and reasoning. The occipital lobe is important for vision and visual perception. And the hindbrain regions, cerebellum for muscle control and balance, and the brainstem for functions such as breathing and swallowing. So the cerebrum is the largest part of the brain, and it is covered by the cerebral cortex. It is divided into the left and right hemispheres. The cerebral cortex, that's the outer layer of the cerebrum, the part of the forebrain, most recently developed part of the brain in terms of evolution, and each hemisphere has its own cortex, like we discussed from the last slide. And the somatosensory cortex processes information about body sensations. The motor cortex processes information about voluntary movement. And my favorite, the association cortex, is the regions of the cortex that integrate motor and sensory information and have a role to play in linguistic and perceptual functioning. And it's interesting to think about the brain in terms of gray matter and white matter, and how gray matter is made up of mostly the cell bodies of neurons, and the white matter are the axons, those fibers, the white matter tracks. So white matter is axons of the neurons, and gray matter consists of those cell bodies. And just a reminder, subcortical means under the cortex. So let's talk about the hemispheres. So our brains are divided into left and right hemispheres. And then each hemisphere is further divided into 4 regions, the 4 lobes of the cerebral cortex. While there's evidence of lateralization or specialization of function, there is no scientific basis for ascribing those functions to personality traits. And for example, what I'm talking about is, which most of you have probably heard someone say at some point in time, I'm more right brain than left, or she's far more left brain than right brain. But there's no scientific basis in that. Unfortunately, we can all consider ourselves to be whole-brained. So, but that being said, there is some evidence for specialization of function. And with the left brain, the left hemisphere, being known for functions such as speech, grammar, and syntax. And syntax are the rules for combining words into phrases and sentences. And then the right brain, right hemisphere, is lateralized for nonverbal information, spatial perception, visual recognition, emotion, sense of humor, interpreting story meanings. And there's a really cool structure that's in between the hemispheres, allowing them to communicate. And it's called the corpus callosum, which is basically a large bundle of axons that connect the brain's two hemispheres. And it relays information between both sides of the brain, which is good because the right hemisphere receives information from only the left side, so it's actually crossed, and the left hemisphere receives information from only the right side, which is why if someone has a stroke in their left side of the brain, they can no longer move the right side of their body, depending on how bad the stroke is, of course. So cutting the corpus callosum seems to leave people with two minds, as the hemispheres are no longer able to communicate. And there is a condition, epilepsy, which is caused by electrical brainstorms that flash across the corpus callosum. So here's another view of the corpus callosum, and we'll have trivia time. So first, how many axons are in the band that connect the two hemispheres together? I'll give you some time to think about it. That's right, 80 million. Number 2, what would happen if a brain surgeon accidentally severed a patient's corpus callosum? Well, they would have a condition called split brain. Very interesting, something very interesting will happen. They will basically have two brains inside of one, and I'll show you a video right after this. And lastly, what medical condition has been treated by severing the corpus callosum? And is that procedure still performed? Yes, it's still performed, and it is a severe epilepsy is the condition that would prompt a neurosurgeon to cut into or sever a person's corpus callosum. All right, and here is the very, very cool video about split brain called Do We Have Two Minds? I hope you enjoyed it as much as I do. So the last section is about brain damage, plasticity, and repair. So neuroplasticity is evidence of the brain's remarkable ability to adapt. Brain damage can produce horrific results, including paralysis, sensory loss, memory loss, loss of speech, also known as aphasias, and personality deterioration. But when damage occurs, can the brain recover its function? And research is finding yes, the brain can recover function in 3 ways. It can recover through collateral sprouting, substitution of function, and neurogenesis. Collateral sprouting is where the axons of healthy neurons that are next to damaged ones grow new branches. And substitution of function describes the process by which the damaged region's functions are taken over by other areas of the brain. And neurogenesis describes the process where new neurons are generated. And here's a video that I've added introducing concussion, which I hope you find interesting. And then to follow it up, I've done some concussion research and found that having more concussions was related to having lower GPA. And if you're interested, here's a little excerpt from my research. Thank you very much.